Hello, ladies and gentlemen. How are we doing today? My name is Richard Murphy, and I'm a second-year PhD student in entomology. And today I'm going to be discussing my paper, which is Segregational Drift Constraints of the Evolutionary Rate of Prokaryotic Plasmids by these fantastic authors right here. And this is going to be in pertaining to mutation and variation, which was the week we had to cover. And I apologize for not being able to give it in person. However, I had circumstances, and hopefully this will do via Zoom, and you'll be able to go back and watch if you've missed anything. So without further ado, let's get straight into it with a little bit of background information. Alrighty. So background information. So plasmids, that's essentially what we're talking about this whole paper. And what exactly is a plasmid? Well, as termed in this paper, a plasmid is extrachromosomal genetic elements of DNA that is capable of replicating independently of the host chromosome. Now, most of us know plasmids as circular DNA within bacteria or prokaryotes that typically carry out a variety of functions, such as employing types of resistance like ampicillin, kanamycin, chloramphenicol, essentially giving the cell some type of benefit over its environment. Now, naturally, it's very selective. However, in this paper, we'll see they wanted to do non-selective pressures. That way, nothing in particular occurred. So in doing this, you may have plasmids that have different allele frequencies or alleles that make up the plasmids. They could be either homogeneous, which is plasmids with the same alleles, or they could be heterogeneous, which is plasmids with varying alleles, also termed as heteroplasmy within this paper. Now, one interesting thing about plasmids as well is that they have varying copy numbers. And this plays an important role in mutation, right? Because as we all know, when DNA replicates or, you copy from one to the next, anytime DNA is replicated, you can have a mutation at a particular base pair or anything like that. Now, with plasmids, for example, they can have a copy number of anywhere from 1 to 15 in most normal cases, or in some cases, depending on high copy plasmids, you could have up to 200 different types of copy numbers. Now, in talking about this, we also need to discuss segregation. Now, segregation of these extrachromosomal element alleles occurs at cellular division. So for example, when the prokaryotic or the bacterial cells begins to divide, that's when the segregation of these plasmid and their alleles, where are they going to go? So this is where recombination or variation may occur, especially in the case of heterogeneous plasmids within a prokaryote. This is because heterogeneous plasmids are going to have varying alleles. So when things start getting mixed around, you can have variation or different rates going on into the future generations. Now, segregation of plasmids into daughter cells. So for example, going from one prokaryote into the daughter cells, and how those plasmids split and go into those cells, is believed to vary upon plasmid localization within the cell as to whether or not, and for example, whether or not the plasmid is clustered or randomly distributed. Now, what this means is, let's say you have a bacterial cell and there's plasmids within that cell. If those plasmids are localized and there's a cellular division, then those daughter cells, for example, one daughter cell might have the plasmid while another might not. Now, if there's multiple copies of these plasmids, imagine if, let's say, it's clustered or grouped in one area, you might have, for example, cells which have varying different numbers of these plasmids, or if they're heterogeneous plasmids, various combinations and different numbers of plasmids within these daughter cells. And so they believe, like I said, that it could be a factor of localization, so the assortment of where these plasmids are located within the cell. And this is going to be something that's going to be studied in this paper, because essentially they're going to study the effects of segregational drift. They're going to compare the dynamics of plasmids and chromosomal alleles under non-selective conditions. This is because typically in nature, plasmids are highly selective. And this is going to increase their evolutionary experimental approach. What we also see here to the right is an example of lateral gene transfer or horizontal gene transfer, which is the byproduct of conjugation within bacterial cells, as we know. So, for example, what we see here in number 1 is we see a simple prokaryote here with 2 different little plasmids in there. Our blue plasmid here is what we're going to see doing the actual conjugation. So as these 2 independent prokaryotes come together, we'll see here at step 3 that once they begin to touch, this transfer of DNA material and transfer of this plasmid is going to occur until we see step 4, where conjugation has risen and both cells are going to now have the plasmid. Now, in this case, we can see it's localized towards the right side. Now, that might just be for the purposes of the drawing or whatnot. But let's get into it. So how exactly did they go about testing their design? Now we're gonna touch on this heavily before I show you the image, and then we're gonna move through to the next figures. So let's first talk about their experimental design because it is very clever. So in order to assess plasmid allele dynamics, the authors utilized a PTAD plasmid, which came from a PWH1277 strain, essentially. Now, what's cool about this plasmid is that it has unknown segregation, right? So people really don't know how the segregation unfolds or synchronization. And the plasmid has no active partitioning mechanism. What this does is it allows for unbiased localization within a cell. Because they know that this plasmid is not going to localize, let's say, next to this region of the cell or next to this or that in the cell, it allows for it to be unbiased. The plasmid could be on your right side, your left side, up here, down low. Your plasmid can be anywhere because there's no partitioning mechanism. Additionally, the plasmid is going to have, you'll see right there, a TA system. A TA system is a toxin antidote system. Essentially, what that does is a toxin is going to be produced, and then likewise, an antidote is also going to be produced. What this does is it tells the cell that it needs to keep and hold on to this plasmid, otherwise it's going to die. Because as this toxin is being produced, it's going to likewise need to produce an antidote. So it's going to say, hey, we got to keep this cell and we got to keep it going, or this plasmid, I should say. We got to keep it going. So the toxin antidote system is very smart because it's going to ensure that the plasmid is not going to be dropped, lost, or kicked out. And remember, they're trying to focus and study on this plasmid. So with talking about this plasmid, the plasmid also encodes for GFP, or green fluorescent protein, under the expression of a LAC-L repressor. Now, essentially, what the LAC-L repressor is going to do is it's going to allow for constitutive gene expression of the GFP, or continual expression. And GFP, green fluorescent protein, is a way to visualize a cell because it, like I said, it's green and fluoresces. So if you use a UV light or a fluorescent type of light, you can clearly see, okay, this cell has this plasmid because you can see it irradiate due to the GFP. And the LAC repressor is going to say, hey, keep producing this, keep producing this. So that's how you're going to see that. So it's a good selection mechanism they have. Now, what they wanted to do was for this experiment, when they have conjugation and these cells come together, and their model organism that they're going to introduce these plasmids to is the Acinetobacter bailey. That's going to be the model organism that's going to have these two variations of the plasmids that they have. So their ancestral strain, which is what they're going to look at as, their original, their ancestral, it's going to have their GFP encoded. And on the sides of the GFP are going to be two regions that are going to encode for NPTLL, but they're going to be broken, they're fragmented. And I'll explain why that is in a second. So these two NPTLL fragmented regions are, if it was, let's say, complete, they would encode for kanamycin resistance. So what they're going to do is, with these bacteria, they're going to introduce plasmids that have the ancestral, which is just the GFP, and the broken fragments. And then they're going to have their novel plasmid, which is essentially going to represent their mutation or what's new being introduced to the population. And this plasmid is going to have NPTLL, which encodes for the kanamycin resistance, and it's fixed. It's the fixed strain. So essentially, if this bacteria has these NPTLL that is functional, then you will see kanamycin resistance. Also another selection mechanism. So the reason they're doing this is because, like I said, the ancestral GFP locus is flanked by two non-functional regions. So if recombination occurs and they come together, then what you could have is that GFP, which is flanked by functional regions of NPTLL, which could then be termed as their heterozygotes, right? Versus their homozygotes, which is going to be the initial GFP tags. All right. So in going with that, we're going to move on to the next thing they have. So like I said, this is a lot, and then I'll show you a visual representation. So with this recombination, it creates heterogeneity as well as the plasmids due to the, like I said, the functional or non-functional addition of the NPLL from the novel strain, novel plasmid, which actually encodes for it. Now, in addition to this, to add to their experimental design, they added two variants to the plasmid, a wild type LAC-L repressor with a tetrameric protein complex that binds to the LAC operon, which I'll tell you in a second what that means. Essentially, what this does is it creates a dimer that binds plasmids together. So this tetrameric protein complex is going to bind two together, which is going to allow for a clustering or a clustered assortment of these plasmids. So as we talked about in the previous slide, if there's clustering that occurs, that could affect how the plasmids segregate and how the daughter cells interact with them. And what they did is they labeled this as their plasmid PTADC, which as you can see is clustered. So it's going to bind two plasmids, tetrameric, Or what they did is they have a PTADR or random, and it's a lac repressor that has a dimeric repressor on the protein, which essentially allows it to only bind to one lac operon, resulting in random distribution of these plasmids within a cell. Because they're not binding to two, they can be free floating, they can be anywhere within a cell, and that allows for a random assumption. So now with all these factors, let's bring it together and see what happens. So what they did with this first experiment was they introduced the novel plasmids, and they had over 23 transformations, or sorry, let me rephrase that, 23 transfers. This allowed for over 160 generations, okay? Also, I must add too that in terms of the copy number of this plasmid, the copy number for the ancestral, because remember there should be more, is 15, and the copy number for the ancestral, which is the new thing being added, is going to be 8. So those are the difference between the novel and the ancestral plasmid. So what they did is they had 23 transfers allowing for 160 generations, and the phenotype was assessed via kanamycin selection of the fluorescence, as well as checking for the ancestral trait too. And what we'll see is we will see 3 outcomes. You either have a fixed novel system, you have a heterozygous novel, which has the heterozygous novel as well as the GFP, or you'll have a homozygous, just your ancestral GFP with the broken fragmentations on the side. And what does that look like? This is our first figure, and this is exactly what it looked like. So we can see here the addition. This is going to be our plasmid right here. We can see our GFP region flanked by the NPTLL regions. Now remember, in our ancestral trait, these are going to be fragmented or broken, while in our novel plasmid, they're going to be functional. We have our lac repressor, and we have our toxin antidote system, as well as a regulation region for this particular plasmid. And then we're going to see over here, this kanamycin resistance, they're just showing it. And then what are our outcomes? Like I said, you have a novel, which has the NPTLL homozygous, right? That's what's being introduced. You have your heterozygous, which could have the novel in addition with the GFP tag. So you'd be able to see that under fluorescence and plating. Then you're going to have your ancestral homozygotes. And then moving on here, so this is your TADR, so the random assortment, and we can just see how, because there's only one binding that happens, there can be that separation, so you can get a lot of heterozygous, which is what they noted, versus clustered assortment, which, as you can see here, there's that binding, that tetrameric binding. So what they notice is they have a lot of homozygote descendants in their daughter cells, splits. And then over here, we just have the number of transfers. Essentially what they said is if we add more DNA, you're going to have a higher transfer rate. And if we have less DNA, you're going to have a lower transfer rate. Okay. And essentially what they did when they did this was they concluded that plasmids assortment plays a role in plasmid evolution on the effects of allele inheritance. The reason how they found this out was because through the random assortment, they found that there were a lot of heterozygotes being formed, while in clustered, they found a lot of homozygous descendants being formed. Now, through both of these systems, though, they did find die-off and eventual extinction, and that's what we're going to take a look at here. So in our second, and let me just go back here one more thing. So after 15 generations, we saw a reduction in novel alleles in the random, so the PTAD random, with an increase in total heterozygotes until the ancestral was extinct, while in the PTADC, we revealed equal homo and heterozygotes due to cluster division in the daughter cells. Now, what we see right here, this is a representation. And the reason they included this was because they had a couple of outliers. So in figure 2, we see group 3, for example, R3 is an outlier because what we see here, if we're looking here, it's the homozygous population. We see that there actually is a homozygous dominance and a hetero-die-off, where in the other cases, we can see a difference where a hetero will continue to go. So what we're going to see is a homozygous die-off, hetero die-off, while in R4 and R8, we see over here, we see the opposite. We show that hetero peaks in capacity and then it dies off. And in all these, they die off starting after the 15th generation, or 15th generation. And over here in B, this is just showing the ratio of heterozygotes to novel homozygotes. And like I said, we see it go down over time because there's a reduction due to eventual extinction. What we see right here, so in this part here, this is through the clustered assortment. So initially it's 1, and what 1 means is there's an equal ratio, so it's 1 to 1, and through die-off that goes down over time. And then over here, we see a number of transfers. So the coefficient of variation, which you would which you would expect to see over time as the variation goes on. And these are the numbers right there. Now down here for C and D. So C is just their way of showing the homozygous or the heterozygous through sequencing and what they could find. So that's how you can see right here whether or not they have, let's say, the fixed trait or not fixed trait. And then over here in D, essentially they did was they ran PCR just to confirm the plasmids. And what they also found was they found something called heteromultipliers. And heteromultipliers are are sister plasmids that can give rise to dimers, so essentially they join together, and higher multimers, and the recombination between non-sister plasmids creates heteromeric or heteromulteric plasmids. So just the joining of together, but that did not affect the transfer from generation. Up next here, this is just showing the proportion of the host and the number of transfers. And like I said, after about that 15th generation, we start seeing extinction. So the die-off, and this is comparing their plasmid alleles as well as their chromosomal alleles, and we see them go down, which is to be expected due to the die-off. Over here, we see the number of transfers. So look, it's proportional because we see as the number of extinctions of host populations increase, we see the number of transfers increase. So the more of these transfers happening, there's actually an increase in the die-off. Okay. And then what we have here, so what they did in and likewise, this is after 160 generations. I just want to add in too for what we're seeing down here. But this is 15th and it goes likewise. So in figure 4, we have a genomic data set survey. And essentially what they did was they surveyed for homozygous plasmid backgrounds. So they went to the plasmid that we had for their PTAD. Where did it come from? Well, it came from, like I said, the PWH1277. So what they did is they did a sequencing analysis or genomic data set analysis to see where did this plasmid come from? Are there any differences amongst the groups? And they surveyed the homologous plasmid backbones. So they looked at the backbones of the plasmids, as well as the derivatives of the plasmids in this study. In order to assess the evolutionary rates, the authors compared the phylogenies of the plasmids and the host chromosomes that they're in. So right here, and what they noticed in A was that they found there was first they noticed that there was an evolutionary change rate. Well, actually, I'm skipping this one thing. Sorry about that. So what they did was they found 2 groups of diverged plasmid sequences separating within the Acinetobacter, which we can see right here. So there was a divergence that occurred. Okay. And this revealed evidence for 2 lateral transfers. However, they do not believe that the lateral transfers were what caused the evolution of the plasmid. They believe it came from vertical inheritance. And the reason they believe that is from what we see in B and C. So this is due to the fact that the derivatives of the plasmids, which we'll see in B and C, we're looking at the chromosomal phylogeny of this. The derivatives, they show a common ancestor, and that's what they're pointing to through both. So there's a common ancestor, so they think it was vertical inheritance that changed this. And the thing about this is what they did is they found that the evolutionary rate of change is slow within this plasmid. And they know this because there's not much variation in terms of the actual sequences between the groups, even in the diverged groups. Essentially, what this is saying is that the sequence was almost the same in both groups. So evolution occurs slowly. And this makes sense with what we've seen back here, because we've noticed that after about the 15th generation, we see a slow die-off all the way down to 160. So obviously what they were doing was, too fast, too much was occurring. So there's a die-off when this happens. And naturally, which is what we see here through, their comparative data set, we see the opposite. We see what we see is the exact thing that they were saying in terms of evolution is slow. Evolution has to be slow. Otherwise, you get these crazy die-offs. So let's go to the conclusion. I've got about a minute left, so we'll finish this up. So conclusion, plasmid assortment affects the dynamics of plasmid allele. We've seen that through random or clustered. In fact, that random typically gives you a lot of hets, while cluster gives you homozygous separation into the daughter cells. Genetic drift during cellular division, segregational drift, leads to rapid extinction of novel plasmid alleles. We see this due to the extinction of both groups throughout the generations as they've gone through, which we can see right there. We've seen them all, they all dip down through extinction. And then we see that plasmid fusion has the capability to be maintained through heterogeneity within a population. This is talking about what we see right over here in terms of those heteromulteric, which are the two plasmids that are joined together. We saw that they were able to be passed on to daughter cells. And then we see, let's see, dynamics of novel plasmid alleles are subject to genetic drift, both within the host and within the population. We've seen this as the cell split. There's varying differences, as well as in the populations and the die-off over time. And segregational drift of plasmid alleles interferes with the fixation of novel plasmid alleles, as there is this die-off that we do see. So segregational drift will have an effect on that. And the phylogenetic reconstruction, which is what we saw in our final figure of the model, reveals that it is a slow evolution of the plasmids gene compared to the chromosomal genes, as seen in figures right here through A, B, and C. And with that, I would like to thank everybody for tuning in with me. We got it in about 20 minutes. I know it was a lot to cover with, there being just about 15 pages of this information. So what I'm going to do is I will open a discussion page. And because I'm not there in person, you guys can ask all the questions that you'd like, and I'll reach back to you in an efficient and timely manner so we can discuss this and I can answer any questions you have. So I thank you for that, and I hope you guys learned a lot from that. Thank you.